Hi, it's an honor to have this opportunity to participate in today's symposium. In this segment, I'll be discussing the fascinating topic of the effects of unilateral gene therapy on the contralateral eye. I'll start by reviewing some of the evidence supporting the existence of the beneficial contralateral ocular effects coming from observations from a number of clinical studies using adeno-associated virus to deliver normal copies of disease-associated genes to the retina. We'll then discuss data from various animal models that may implicate potential mechanisms underlying these contralateral effects. The strongest evidence of contralateral ocular effects comes from the literature on gene therapy for LHON. Delivery of normal human ND4 to subjects with vision loss due to the 11778 G to A mutation has been studied by three independent groups, Bin Li and colleagues in Wuhan, China, Gensight in France, and John Guy and colleagues at the Bascom Palmer Eye Institute. All of these groups have reported some degree of evidence of an effect of gene therapy on the contralateral uninjected eye. In China, there's been 3 investigator-initiated trials and then one phase 1-2 clinical trial of the study drug NFS-01. All 4 of these studies employed unilateral intravitreal injection of the AAV while monitoring the visual outcomes in both eyes. All 4 have demonstrated improvement in the best corrected visual acuity, defined as at least 0.3 logMAR units on the uninjected eyes of 1/3 to 2/3 of patients by 12 months post-injection. In several of these studies, the rates of improvement in the uninjected eye has approached that of the treated eyes. The initial investigator-initiated trial now has 7-year follow-up data for the cohort of 9 subjects. One of them, patient 1, was injected bilaterally, and another, patient 8, did not have data beyond 3 years. However, of the 7 other subjects, 4 of them demonstrated visual acuity improvement in the injected eye at 7 years, depicted in each case with the solid red line. And 5 of the 7 demonstrated improvement in the contralateral uninjected eye, depicted with the dotted blue lines. While no sham controls were included, based on the natural history of the 11778 mutation, these improvements in approximately half of the subjects is strongly suggestive of a beneficial effect to the contralateral eye. In addition, the 3 major trials from Gensight using GS10, which is also AAV-mediated delivery of ND4, all documented improvement in the best corrected visual acuity by several lines on the ETDRS chart compared to the post-treatment nadir, as we had just discussed. And this was both in the injected eye and also to an appreciable extent in the uninjected eyes. As before, the caveat is that there was no control arms in which the subjects didn't receive study drug at all. Recently, unpublished data from 2 investigator-initiated trials in China of the drug NFS-02, which introduces the normal human ND1 gene to those subjects with the less common variant of LHON, also indicates a beneficial effect in half of uninjected contralateral eyes, which is similar to NFS-01. Now, there might be some other examples of contralateral effects of ocular gene therapy. In non-peer-reviewed data from AGTC, from their phase 1-2 study of replacement of the CNG beta-3 gene in cones for treatment of achromatopsia, they've shown that one of their outcome measures, which is the severe light aversion experienced by these patients, there was improvement in both the treated and untreated eyes at 12 months post-treatment. The visual acuity measures in these subjects has not been reported yet. And then in the earlier studies for gene therapy for Leber congenital amaurosis due to mutation of the RPE65 gene, in which the gene therapy was to the retinal pigment epithelium was initially limited to just one eye, there were examples of improvement in visual acuity and visual field sensitivity in the uninjected eye, repeated by multiple groups. On the right, we have early data from Gene Bennett's group at University of Pennsylvania, which shows the longitudinal changes in visual acuity in individual subjects in a phase 1 dose escalation trial. In this case, a negative change in logMAR represents visual improvement. While the injected eyes more consistently showed improved visual acuity, there were a number of examples of improved acuity in the uninjected eyes as well, such as this subject in green here. So there does seem to be reasonably solid evidence that genetic manipulation of one eye can impact the fellow eye in heritable retinal disorders and optic neuropathies. The question is, what is the mechanism that could explain this salutary effect? Well, it's possible that some type of psychophysical effect might be an explanation by increasing the overall input to the visual cortex. It would seem that the most likely explanation would involve some element of material transfer from one eye to the other. Now, this could involve transfer of the viral vector itself, such that viral DNA ends up transducing retinal cells of the fellow eye. Another possibility is that there could be a transfer of the product of the gene therapy, such as uptake of viral RNA transcripts, which are then translated and subsequently incorporated into the mitochondria of the fellow eye. Or it could be that the modified mitochondria from the retinal ganglion cells of the injected eye ultimately find their way to the retinal ganglion cells of the uninjected eye. Now, where might this transfer occur? It could potentially be systemic via the circulation, especially in the case of the viral vector itself. Or it could occur at the level of the optic chiasm, where retinal ganglion cell axons from each eye are coursing past each other in close proximity. In this location, there could be a direct transfer between neurons potentially, or glial cells could be acting as an intermediary, receiving mitochondria or viral products from one axon and transferring it to another. I'll be discussing some animal models in which AAV injections are performed in one eye, and then the presence of viral nucleic acids is assessed in various ocular tissues of each eye over time. I'll also discuss some mouse work in which the roles of intraocular pressure and the gap junction protein connexon 43 are investigated in this process. Now, in studies of both NFS 01 and GS10, non-human primates underwent unilateral intravitreal injection of the study drugs, and in both cases, viral DNA was detectable in ocular structures in the contralateral eye of some of the animals at 3 months post-treatment. And that's shown in orange in each case, whereas the blue represents the injected eye. Excuse me. However, in the GenSight sponsored study, the level of viral DNA that was found in the contralateral retina, which is where the cell bodies of the retinal ganglion cells reside, was nearly 3 orders of magnitude less than in the retinas of the injected eyes. Furthermore, in a follow-up study, it was reported that the RNA transcripts of the viral-delivered ND4 were not detectable in either optic nerve or retina of the contralateral eye at 3 months. And at 6 months, neither viral DNA nor RNA transcript were detectable in the retina of the fellow eye, whereas they remain easily quantifiable in the retinas of the injected eyes. So this raises significant doubt that viral transduction of retinal ganglion cells is occurring in the fellow eye at a sufficient rate to produce a positive therapeutic response. However, in a mouse model, there was some interesting data from a very recent publication that in mice treated with AAV ND4 vector, the context of elevated intraocular pressure seemed to affect the rate of detection of viral RNA transcript in the fellow eye. They achieved this by doing an anterior segment injection of microbeads to raise the pressure. So they found about a third of the mice with elevated IOP had viral RNA transcript detected in the contralateral retina, whereas with normal IOP, it was not detected in any of the subjects, albeit with the caveat that their sample size was only 4 in this case. More interesting is that among the mice with elevated IOP, viral RNA transcript was not detected in the contralateral retina if the gap junction protein connexin 43 was deleted from astrocytes in a cell-specific manner. And so this raises the question of whether astrocytes at the optic chiasm might serve as a conduit for material transfer. I'd like to close with some extremely preliminary, but I think very interesting data from my own lab, where we've taken a recent interest in the contralateral effects of unilateral gene therapy. So we hypothesized that there may be exchange of intact mitochondria between RGCs at the level of the optic chiasm. In a pilot experiment that we recently did, we injected mice in one eye with AAV carrying a construct that encodes green fluorescent protein embedded in the mitochondrial outer membrane. While the efficiency of the viral transduction in this experiment left a lot to be desired, you can see that there are a number of GFP positive cells in the retinal flat mount from the injected eye, with the RGCs highlighted with the marker RBPMS in purple. Within individual cells, those that were transduced did express the construct at a very high level, and you can visualize mitochondria in the RGC axons as they're coursing towards the optic nerve head. Now, interestingly, when we examined the contralateral uninjected eye, we observed patches of retinal ganglion cell somas with GFP signal, which is a potential indication of successful interocular transfer of mitochondria. Now, we're in the process of assessing the replicability of this finding and also performing some key controls. But this observation is intriguing and might support the idea of bulk transfer between the eyes. So to conclude, contralateral effects of gene therapy in uninjected eyes have been observed in trials treating LHON and other retinal diseases. Animal models have shown evidence of varying levels of viral DNA and RNA transcripts in contralateral ocular tissues. This material transfer of nucleic acids might be impacted by intraocular pressure of the injected eye and by the presence of gap junctions between astrocytes and neurons. And the transfer of modified mitochondria between eyes might also be a possible mechanism for contralateral effects of gene therapy. Most importantly, the possibility of contralateral effects must be acknowledged when designing gene therapy trials, and especially should impact the selection of appropriate controls. Thank you for attention, and I'm happy to take questions during the breakout session.